Swayam Prabha. Digital India. Educated India.
Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. In the previous class we have been looking at the copper proteins, and the class prior to that we looked at the type 1, and in the previous class we have again looked at the type 1, how the electron transfer takes place, and then we also looked at variety of type 1 copper proteins having different redox potentials starting from 180 to about 680 millivolts. Then we looked at the type 2. There are two examples that we have looked at the type 2. One is galactose oxidase, other is amine oxidase. In both the cases there are two half of the cycles. In the first half the substrate is converted to the product. In the second half the converted enzyme is regenerated back to the normal, and that is what happens in both the cases. And so therefore, and to regenerate back you use the oxygen. That means enzyme is in reduced form back to the normal form. So, that requires the oxygen to get reduced. So, though the oxygen is not directly involved in the initial reaction of conversion of the substrate to the product, it is involved in the second step where the converted enzyme, the reduced enzyme to regenerate back to the normal in order to continue with the cycle. So, this is true both with the galactose oxidase as well as with the amine oxidase. Then towards the end of the previous class we looked at another topic on melanins. Melanins, as you know, that it is from the term comes is a black kind of a color, which comes from the oxidation of tyrosine by variety of enzymes, and this leads to a pigment, black pigment. In fact, this is the black pigment which basically dissipates all that UV radiation that falls on the skin, and thereby it protects a person from attacking by the cancer kind of a situation. And this is what where the skin where there is no tanning, they try to go for tanning, so that the tanning will help them from the sun radiation, particularly in the UV region of the sun radiation in that. So, we have looked at also this enzyme. The enzyme is called tyrosinase. It is a type 3 copper enzyme, and I will be telling you an example little later. Here we will look at how the tyrosine is basically converted through a variety of reactions to melanin kind of thing. So, a lot of redox processes goes on. Okay. So, the tyrosinase can convert into dopa-quinone, and so also L-dopa can be converted into dopa-quinone as well, and these can internally convert the cyclodopa and the dopachrome. So, these dopachrome in turn connects by a variety of enzymatic reactions, including the tyrosinase, to give the eumelanin, which is a polymeric form of this, and that is what generates the black color. Now, let us look at an enzyme on tyrosinase. Tyrosinase has got a type 3 copper center, and this is a 2 copper centers present together. This particular tyrosinase, we have a tetrameric structure, as you can see, and each of this has the reactive center. The reactive center is shown over there, where the these green balls correspond to the 2 copper centers, and no copper-copper bonding or no copper-copper connectivity at all. Both the copper centers are present in copper 1 state, and both the copper centers are connected to the histidines. So, therefore, you can see from here a copper with 3 histidines, a copper with 3 histidines, and no copper-copper binding in this. So, this is in the copper 1 state. So, in this enzyme of tyrosinase, so let us look at how the tyrosinase functions. So, before that, let us have a look at when you have a protein in the resting state is the dicopper with the 3 histidines on each and no connectivity between the copper, and each one is a copper 1. Now, when you add the oxygen molecule to this, and each of the copper 1 can donate 1 electron and go to copper 2, therefore, totally 2 electrons can be donated, and when the 2 electrons are donated to the O2, it becomes O2 2 minus, and that is what you see here, a bridged peroxo kind of a species, and such kind of thing that we have already seen earlier when we were talking about the oxygen transport proteins, hemocyanin. Okay? And this can be identified from the infrared spectral vibrational frequencies of OO stretching vibrations. Now, let us come to the mechanistic aspects of the tyrosinase, and in the free enzyme, which you call it as a deoxy form of it, the copper 1 center, and since it is a copper 1, it can readily react with the oxygen, and then this oxygen will get converted to O2 2 minus, as we have shown here. It is the same, and this O2 2 minus is bridged between the 2 copper centers, and now copper centers are, each of the copper center is copper 2 oxidation state, not anymore in 1, because it donates 1 of the electron from each of the copper center. Now, this upon binding to the substrate, in this case let us take the substrate as the catechol, and the catechol can bind very nicely to the dicopper center, and this upon the protonation can lead to the oxidized form of the catechol, which is called catequinone, and the other the oxygen goes as a water kind of thing. So, you can see a connected species, still one more oxygen is there, it is a connected species. So, therefore, this species is still active, therefore, it can interact with the second molecule. So, this is one of the rare enzymes where the enzyme is regenerated back after doing 2 sets of or 2 cycles of 2 sets of reactions. So, you have first one reaction here and the second reaction. So, another catechol molecule will be bound here, and this will be converted to the catequinone and then back to the deoxy. Okay. So, you understand this, the copper 1 state becomes O2, 2 electrons coming from each, O2 2 minus, which is bridged between them, and this is sufficiently good enough to oxidize this, and then in the presence of the protonation you will get the catequinone plus the water. That means one more oxygen of the O2 is still left over, and that is what you see as a bridge species, and this form of the enzyme is still active, can bind to one more mole of the catechol, and this will be binding to this, and then finally you get totally 2. On the other hand, instead of the dihydroxy compounds, if you have a monohydroxy, which is nothing but phenol. See here the active species, and this in presence of the phenol can bind. Since phenol has got only one hydroxyl, it can only bind monodentate, while the catechol can bind bidentate because of the 2 hydroxyl groups, and this will further oxidize this one, and that will release the catequinone. So, of which one of the oxygen is coming from this O2, and the second oxygen goes as a water. So, the same enzyme, if it is a di-OH compound, diphenolic or catecholic type of a compound, you have one water molecule and it will do a second cycle of the catechol. On the other hand, if it is a monophenolic kind of a situation, both the oxygens are used. One oxygen is used to convert the phenol into catechol, second oxygen is used converted into water, and no more second mole. So, therefore, this is what the difference between a monophenolic or monohydroxy compounds versus dihydroxy compounds. I hope you understand. Type 3 is a dicopper center. There are cases where the enzymes have got type 1, type 2 and also type 3 in the same enzyme. So, we will see a situation of that. Example, here we are looking at ascorbate oxidase. Look at the ascorbate oxidase. So, this is the ascorbic acid and goes to dehydroascorbate. So, these are all steps that you can try to look at, understand that the deprotonation at pH, and then one more deprotonation and the oxidation, and one more oxidation. So, you need a dioxidation, obviously you are getting 2 keto moieties here and from the 2 phenolic moiety 2 hydroxy moieties over there. So, ascorbic acid, dehydroascorbic acid. So, this is basically a oxidative reaction. So, therefore, the enzyme is a redox kind of an enzyme, and it acts on the diphenols, dihydroxy compounds we have also seen in the previous example, and they are as donors and the oxygen as acceptor, because oxygen will accept the electrons. Okay. So, L ascorbate plus O2 giving 2 dehydroascorbate plus 2 H2O. So, that means your oxygen what you added is not incorporated into your ascorbic acid, both the O's are gone into water. We have seen in the initial cases monooxygenase, where one oxygen is used and the other oxygen goes into water. We have seen dioxygenase, both the oxygens are used and no water, and here is an example where both the oxygens are unused, but oxidation takes place and this goes into the water. Both the oxygens go into water. Okay. So, L-ascorbate oxidase is an enzyme that catalyzes the ascorbic acid into dehydroascorbate, and this is the 2-dimeric enzyme that you can see. And there are 3 different types, as I said. Type 1, which is blue copper protein; type 2, single copper, but is a different kind of a one; and type 3, which is a dicopper center, which is involved in redox process. Now, you can see this here. This is the type 1, which is a blue copper protein, which is connected by 2 histidines, 1 methionine, 1 cysteine. You can see here, and you can see here this is the type 2 copper center, and you can see here the 2 coppers, which are the type 3 copper center. So, there is a distance of about 15 to 20 angstroms from here, but these are within 3 to 4 angstroms distance. So, because these are within 3 to 4 angstrom distance, the type 2 and type 3 join together and jointly do a catalysis, and whereas type 1 is involved in what? What did we study for the type 1? Type 1 is a blue copper protein which is involved in electron transfer. So, therefore, it is basically electron transfer kind of a reaction. So, this will do a part of the electron transfer, this will try to hold activate the oxygen, and in between you will have a substrate, and the substrate will get oxidized, and that is how the whole reaction will go. Now, let us look at the sequence of the electron transfers in this. Let us call type 1 as T1 and type 2 as T2 and type 3 as T3, and in the bracket it is shown some value. For example, here 0, that it is to be taken as not 0, it is to be taken as oxidized kind of thing. So, T2 oxidized, T3 oxidized, T4. So, all the 4 coppers are in oxidized state. Now, if you add 1 electron to this particular enzyme, where will the electron go? Will it go to T1, will it go to T2, will it go to 3? There are everywhere you have the copper 2, but it will go only to T1, because the redox potential of this is very favorable. Therefore, that will attract the electron. So, that will go to T1. The second electron will go to T2. Now, you have T1 is in the reduced state, T2 is reduced state. Now, these 2 electrons are internally transferred to T3, and both the coppers of the T3 will get reduced. What is the necessity? The necessity of the dicopper center is because it can capture the O2, it can release the 2 electrons, 1 from each, and convert the O2 into O22 minus. We have seen already. That is mainly the reason why the electrons are transferred from T1 and T2 to the T3. T3 has 2 coppers, so it requires 2. So, the next step is T3. That means now the enzyme is ready to activate the oxygen. So, at this stage, 1 more electron reduction will make 3 electrons, and the 3 electrons will take up the oxygen moiety into this, okay, and that will be activated to throw out water, and one of the O is bridged. So, this is out of the 2 oxygens, 1 O is bridged, 1 goes as a water in presence of the protons. Already 1 is gone. Now, so now what it requires? It requires only 1 more electron, because O2 has got 2 bonds. Okay. So, therefore, 1 more electron will be reducing that oxygen again into the water. So, you get 2 water molecules effectively, and the substrate will get accordingly oxidized. So, the substrate will be in this particular region, and as you can see, the substrate goes from here and the oxygen goes from here. Okay. So, therefore, you have a substrate getting oxidized and the O2 getting reduced, and the O2 reduction requires 2 coppers together. So, I think the whole thing is understandable now. First electron going into T1, second electron going into T2, both the electrons going into T3, and in presence of one more electron, 3 electrons will reduce the O2 to water and the abridged oxo species. The nature of this is not very well known, and this with one more electron will ooze out the second water as well, and the substrate gets oxidized. So, as you can see that. So, therefore, you can see this is your ascorbic acid, dehydroascorbate, that means water is gone, and now you can see that the kind of a copper centers here, you have 2 other copper centers here and one is this one. So, therefore, these are, you have all of these are over there, and these are your type 3 kind of things and type 2, and these are roughly within this kind of a triangular shape. Therefore, O can be activated, O2 can be activated between the 2 copper centers of type 3, and then at the end one O can be bridged here, and that is what you can see. So, that requires the arrangement of the 3 coppers in a very close fashion, and that is what you would see there. So, substrate, organic, electron transfer kind of thing, and so this is the kind of a redox reaction that happens. So, I hope now you understand the type 3, we have seen tyrosinase, we have also seen a case where both type 1, type 2, type 3, all 3 are being present, and these oxidize ascorbic acid into the dehydroascorbic acid to hydrogen hydrides are lost, so therefore oxidized. Now, one last enzyme that we would like to look at is very important in the body, because body has always has an assault by the oxygen radicals, because the cells work on oxygen. So, therefore, oxygen radicals have been continuously formed, and these oxygen radicals are toxic. So, therefore, there should be something that evades these, removes this. We have already studied a lot on superoxide dismutase in the iron and manganese and other enzymes. So, now we look at the copper case. So, the general thing is oxygen going to superoxide, and this can be dismuted by the superoxide enzyme, dismutase enzyme into peroxide and water, and this can be converted, this in presence of the metal ions can convert into radicals. These radicals are very bad for the cell tissue can get damaged, DNA can get damaged. On the other side, you can see the oxygen with the superoxide, copper zinc superoxide, that is what we are going to now study, and this will convert this into the H2O2 plus water. If you have a catalase that can further degrade H2O2 to H2O and O2. So, so SOD and catalase together can completely remove the superoxide species into that, and as long as there are no iron or metal ion species which undergo redox, it can be iron, it can be even copper. If it can undergo redox metal ion, then that can make the radicals. This is what is known as the Fenton reaction. So, if such a thing is there, it is dangerous. Okay. So, in absence of this can take care into the oxygen into water, and as you can see, it can get into the mitochondrial system, nucleus, all this can get damaged. Now, let us look at this last enzyme, which is superoxide dismutase. Superoxide dismutase is a copper and zinc bimetallic. Of bimetallic, one is a copper, other is a zinc. Okay? And this is present in the human, and this is a homodimer around 32 kilodalton. Okay, it has an intramolecular disulfide bond and a binuclear copper zinc site, and this copper zinc site is absolutely necessary for the catalysis for the disproportionate of the hydrogen the superoxide into hydrogen peroxide and dioxygen. So, if you do not have this, no reaction. Of this, the copper is the reactive center, catalytic center, and the zinc is the structural center. Suppose if I replace zinc by cobalt, zinc by nickel, still we can have the activity. But if I replace the copper center by something else, no activity, the activity drops down dramatically. So, in fact in the biological system, the copper chaperones SOD1, they facilitate the copper insertion, and they also cause disulfide oxidation, okay, to form structurally form this one. So, the mature protein is highly stable, but unstable in its metal free and disulfide. If it does not have a disulfide, the protein is not stable. If it does not have a metal, it is not stable. So, in the superoxide, we have seen several times this reaction O2 minus dot plus O2 minus dot plus 2H plus gives H2O2 plus O2. So, it is a dimer, one here, monomer here, and there is a 2 metal ions, one zinc, one copper, one zinc, one copper that you have. And how is this bridged? Unlike the previous case, 2 copper centers, nothing bridging in case of type 3, but there is a bridging here. You see that bridging is nothing but imidazole, one nitrogen is towards the copper, other nitrogen is towards the zinc. So, that means these two are well connected, and this is the one, this is the histidine imidazole bridge between the two centers. So, one is, this is the zinc center, this is a copper center, copper is a catalytic center, zinc is a structural center. Structural center can be replaced by other kind of ions, but catalytic center cannot be spared. So, now, so you take the native form of the enzyme where you have a copper 2, and now you add the O2 minus, the O2 minus will bind at the copper 2 center, and the redox takes place and it will go as the O2. So, that means it takes the electron out and copper 2 becomes copper 1, and the O2 minus dot becomes O2, and the O2 is out. Now, this is ready for the second mole of enzyme, that will make a disconnected kind of thing. Second mole of O2 minus dot is bound again at the copper 1 center, which will again reoxidize this one, okay, and that will replace, that will go to the H2O2 and will go back to the copper 2. So, in one the copper is reduced, in the other copper is oxidized. Okay. So, similarly, the O2 minus and is oxidized and the O2 minus is reduced. So, O2 minus oxidized means O2, O2 minus reduced means H2O2 or O2 minus. So, the first step you get the O2, the second step you get H2O2. So, you could see how the enzyme is undergoing from copper 2 to copper 1 and copper 1 to copper 2, the O2 minus to O2 and the O2 minus to O2 minus. So, this is the kind of thing that we have in all these that okay. So, we have seen quite a good number of examples of all these, as you could see that the how a type 3 copper reacts, type 3 copper because it has the 2, yeah, this is also type 3 copper, tyrosinase continuously bringing redox process on the tyrosine molecule, then end up with the melanin that we have looked at. And actually the tyrosinase enzyme having 2 copper centers, the 2 copper centers can convert the biphenolic compounds or catechol like compounds, 2 moles will be reacting. So, the first reaction will give the catechol, second reaction will give another catechol, and so in each case the oxygen is converted into water, totally 2 equivalents of the water is converted in all this. And so, whereas in the monophenolic case only one equivalent is reacting and that gives the catechol, and there is that you have seen. So, ascorbate oxidase from ascorbic acid to dehydroascorbic acid, as you can see, and this is the dehydroascorbate. So, this is again an enzyme where the type 1, type 2, type 3, all 3 are present, they work very harmoniously, and type 1 to type 2, type 3, you can see that this particular thing distance is around 15 to 20 angstroms, and whereas the type 3 to type 2 is around 4 to 5 angstroms. So, therefore, type 2 and type 3 together activate the O2 and this one do the redox process, and overall the substrate is oxidized, O2 is reduced, and if the substrate is already diphenolic or di-OH, then both the O's will go as water and that is what you see in the dehydroascorbic acid. And this is where I explained to you that they all three together sitting over there as a coordination. And the last part, as we have seen, that it is a superoxide dismutase. We have already studied this kind of an enzyme several times, and this has a bit different from what we have studied in iron, what we have studied in manganese, etc. Here you have a copper and zinc. So, copper and zinc are connected by the imidazole, and that imidazole part is the one which is bridging the two things. And here what happens, the one O2 minus is oxidized to O2, that means copper is reduced to Cu1, and the second mole is of O2 minus is further reduced to O2 minus, that means copper 1 is oxidized to copper 2, and that is what the mechanism is. So, in effect, under this particular story of copper, bioinorganic chemistry, biological inorganic chemistry, or inorganic chemistry of copper in life, we have looked at very detailedly type 1, which is a blue copper protein. We have looked at the type 2, which is involved in the redox process like galactose oxidase, amine oxidase kind of things. They do not use the any oxygen of the O2, but the O2 is required to bring back the enzyme to the normal state, because all these reactions go in two halfways. The first is substrate is converted and enzyme is reduced. The second step, in presence of the O2, enzyme is reoxidized. Type 3, we have seen enzyme tyrosinase, which is involved in the melanin formation, tyrosinase which is involved in the bi-catechol kind of thing, phenol kind of things, and the two copper ones can play a role to copper 2 and back. Type 4 is the one we have already seen. Type 4 is nothing but type 1 plus type 2 plus type 3, all the three together. So, this is the type 1, these two are the type 3, and this is the type 2. Okay. So, the substrate comes here, oxygen goes here. So, you have a mechanism to open and close, open and close. And copper A, copper B we have not looked at, copper Z also we have not looked at, and superoxide dismutase we have of course looked at this superoxide dismutase. We have looked at earlier and we have seen that one too. So, in this particular case, the zinc is the structural, copper is catalytic. If I replace the zinc by manganese, zinc by cobalt 2 plus, zinc by nickel 2 plus, still the reaction goes. But on the other hand, if I replace the copper by some other ion like manganese or zinc or any other thing, the whole reactivity is gone, and that is how you find that the copper is catalytic in nature and the zinc is non-catalytic in nature. So, thus I think we complete with the copper enzymes in this particular class, and in the next class we will look at the zinc based enzymes. Thank you.